The last screencast we talked about focused on functions of the digestive system, components of the oral cavity, including different types of teeth, number and types of teeth. We talked a little bit about the salivary glands. And then if we compare the gizmo to what our notes covered last time, we talked a little bit about the small organs associated with the digestive system, so pancreas, gallbladder, and liver. Today, I want to focus on the large organs as it compared to your gizmo. And so today we will talk about the esophagus, the stomach. Everything inside this box are the small intestines. Your gizmo allowed you to drag and drop separately the first segment from the second and third because the first segment has a much more important role in digestive than the second and third. So that's why they allowed you to drag and drop them separately. Today we'll talk about the large intestines, and that will be our focus for today. So the esophagus, it is located behind your trachea. So if you run your fingers up and down the front of your neck, you'll feel the cartilaginous rings of your trachea. That's your windpipe. Right behind it is the esophagus, and it is probably collapsed right now. Every time you swallow a bolus or a collection of food, then it will open up its lumen and accept that. And then peristalsis, which is the rhythmic contraction of the GI tract, will help to move that substance down the esophagus. We want to move down the esophagus into the stomach. And once you swallow, you no longer are in control of the movement of that food because now we begin to see the segmentation of peristalsis. So the analogy in your book was like trying to squeeze toothpaste out of the tube. And so we can see the rhythmic contraction because remember that the GI tract is made up of 4 layers. And so the lumen would be where the bolus would be found. The mucosa then would be closest to the lumen. The submucosa is holding the mucosa layer onto the surface of the inside of your GI tract. And then there's that muscular layer underneath the submucosa, involuntary peristaltic contractions, which help to move the bolus from the esophagus to the stomach. So the whole function of the esophagus is to move the food from your throat or pharynx into your stomach. You'll notice that there is the diaphragm here. That is an organ associated with the breathing mechanism. We're going to have to pierce through that layer in order to be able to make it to the stomach. At the very end of the esophagus, before it enters into the stomach, is a ring of muscle that is referred to as a sphincter. And that sphincter can open or close the opening from the end of the esophagus into the stomach. Inside of the stomach are going to be gastric juices with a very low pH. And so sometimes that gastric juice can escape back up through the sphincter into the esophagus, and that can lead to a painful sensation, sometimes known as heartburn or acid reflux. And so you can take medicine in order to deal with heartburn or acid reflux. So that is the job of the esophagus. Let's talk about the stomach. Your book describes it as a J-shaped organ. The very top region, there's 3 sections of the stomach, is the fundic region. And so you can temporarily hold food that is going to be processed next there. In the fundic, the cardiac region is just below that, and that is the middle section of the stomach. And then the very bottom section of the stomach is called the pyloric region. So there's 3 sections to the stomach. It's on the left side of your abdomen, so your left side. 10 inches is the length of the stomach. When you look inside of the stomach, you will see rugae. Rugae are folds of the inside of the stomach. And so you can see all the rugae here. That allows the stomach to be able to stretch if you're eating a really, really big meal, like say Thanksgiving. And that allows the stomach then to hold a maximum capacity of about a gallon of food at one time. So you think about a gallon of milk, that's a lot. Once the bolus from the esophagus enters into the stomach's job is to convert the bolus into chyme. Chyme is what is going to leave the stomach and is going to be the absorbable types of food substances that will enter the small intestines and hopefully be absorbed. So the stomach first function is to accept food. The second function is to convert it to chyme. And the third function is to deliver it to the small intestines. Now, in the walls of the stomach are gastric glands. And there are two major kinds of cells of the gastric glands. There are the chief cells, and if you remember from the gizmo, the parietal cells. The chief cells release an enzyme called pepsin, and pepsin helps to break down proteins. Parietal cells release the hydrochloric acid that creates a pH of about 2 in stomach acid, and together we refer to those are as part of the gastric juice. And so that's the stuff we don't want to go back up into the esophagus. So we have a sphincter to control that. Okay, let's talk about the small intestines. Collectively, the whole thing is around 18 feet long. 3 sections, duodenum or duodenum, jejunum, and then ileum. Ileum and jejunum, you were allowed to drag and drop together. The duodenum is about 10 inches of the first portion of the small intestines. And then the jejunum is 7.5 feet on the average, and the ileum is 10.5 feet. And so roughly around your small intestines averages around 18 feet. When you look at the inside of the small intestines, you will see circular folds that will give the inside of the intestines a velvety appearance. And that's what you would be able to see with the naked eye. If you were to take a section of this, put it on the microscope, and look at it on the red or low objective at around 100 times, you could see each of the velvety circular folds. But if you were to focus more on a section of the velvety folds and look at it on, say, the yellow objective lens at 400 times, you would see a single villi. You had a section on your gizmo about villi, or a villus is one. A villi is primarily a blood vessel, a capillary blood vessel, and a lymphatic vessel. And together, that lymphatic vessel and capillary vessel can allow for maximum absorption of food particles, or chyme. But if you were to take a villi and look at it on the microscope, so now we need to go up to, say, the blue objective lens and look at 1,000 times, a villi has its own villi called microvilli. All this is doing, girls, is increasing surface area for the maximum absorption of substances. And so when we talk about the maximum absorption of substances, we're talking about monosaccharides of sugars. We're talking about the amino acids of proteins, and we're talking about the glycerol and fatty acids of lipids. But remember, lipids are non-soluble. They will not go into solution and be absorbed as easily as the carbohydrates and the proteins. And so another function of the small intestines is to emulsify the fats. So here are the fat components. They do not want to mix with water, but bile, which is released from the gallbladder, which was made from your liver, helps to emulsify fats and to allow them to be changed and brought into solution. So once they're brought into solution, they're easily absorbed in the villi. And so those are two important functions of the small intestines. The third is to take anything that is non-digestible and then continue its pathway on to the large intestines. So here we are with the large intestines. The large intestines is going to begin with what your book calls a blind opening. What that basically means is it's like a cul-de-sac. So in from the small intestines comes all of the non-digestible and watery remains of what's happening in your digestive system. At the very end of the cecum, that's what we've got here in the box, is the vermiform appendix. Vermiform means worm-like. Your appendix used to be super important back when you ate other types of rough foliage, and so we don't really have to worry about digesting that anymore. So we don't really need our appendix. It can be removed surgically if it ruptures, because if it ruptures, that is a big deal. Whatever is left in the large intestines will then travel up the ascending colon, across the transverse, down to the descending, where it will go into the sigmoid colon, where it will be temporarily stored until the sensation of needing to have a bowel movement. And when that happens, then it will be removed or eliminated through the rectum. And so that's the rectum. The very end of the rectum is the anus, and there is another sphincter there that allows for the voluntary control of the mass movement of fecal material during a bowel movement. You can see these little pouch-like areas all along the colon. Those are called haustra, and haustra, and they allow for whatever's moving through the large intestines to kind of linger a little bit longer in that space to allow for the maximum absorption of water. And so there is a muscle, tinea coli, that is found along the entire length of the colon, and it contracts in order to help with peristalsis, or the movement of whatever's left in the colon to travel down to the sigmoid and eventually be removed from the rectum. Feces is about 75% water and 25% solids. In that solid portion of fecal material is bacteria. And so the process of releasing that is then defecation. Okay, that's it.